So this session is really to talk about, as this year, as we've known, we've had some setbacks with trials. And so we're going to talk about the status of 3 trials that have been long in coming, difficult in their outcomes. We're going to talk about what we've learned. So the first speaker is Ron Victor. He's going to speak about the Eli Lilly tadalafil study.
Morning, thank you, Pat. So I'm the chair of the steering committee. I'm going to begin with a bit of background about leading up to the trial, because the preclinical and proof of concept data were very strong. Okay, so dystrophin, as you all know, is not just a structural protein, it also is a scaffolding protein. So it brings other partner proteins to the sarcolemma of the muscle membrane, and among those is an enzyme called nitric oxide synthase. There's a muscle-specific form of this, a lean skeletal muscle, and it has to have dystrophin to work properly because it has to be in the right place in the cell. It's quiet at rest, and it's only activated when the muscle's active. The nitric oxide produced dilates the blood vessels. It makes the blood vessels relax, open up, and feeds the muscle with enough blood flow and oxygen to meet the metabolic demands of working muscle. Oops, a little quick on the trigger. So in boys with Duchenne, where there's no functional dystrophin, there's a secondary loss of nitric oxide production from the muscle during exercise because the enzyme that produces it, the nitric oxide synthase, is in the wrong place. It's not at the muscle membrane. So because of that, the smooth muscle and the blood vessels can't relax, the vessels can't dilate, there's not enough oxygen blood flow supply. That's what we call ischemia during exercise. And the question is whether the impaired blood flow regulation would contribute to use-dependent injury. So this is not going to be a mechanism of rest only when the muscle is exercising, and that's really important. Now, tadalafil could possibly boost a residual signal of nitric oxide from the muscle by working on the downstream target called cyclic GMP. And the question is, would a drug like tadalafil, a PDE5 inhibitor, restore normal blood flow regulation, therefore protect the muscles from use-dependent injury when the boys are active during the day and the activities of daily living? So real quickly, this was initial preclinical proof of concept. 1 dose of tadalafil prevented post-exercise fatigue. On the left of the slide, this shows that mice normally in their cage are very fidgety. They stand up a lot, and every time they stand up there's a dot. And there's C57, the normal mice, the MDX, and the muscular dystrophy, Duchenne-like mice. And before exercise, they're both spontaneously active. After a brief bout of exercise, the normal mice back in their cage are still active, but the MDX mouse just sit there as though they're fatigued. And the mechanism seems to be that the blood vessels, the tiny blood vessels in the muscles, go into spasm. And a single dose of tadalafil, a few hours before the stress test, prevents the spasms, and it improves the 6-minute walk test in the mouse. So that's shown at the bottom of the slide. Now, that was 1 dose of tadalafil. Chronic treatment with tadalafil, Cialis, or Viagra, sildenafil, another PDE5 inhibitor, has been shown to benefit both skeletal muscle and cardiac muscle and the diaphragm. In the MDX mouse, with all these papers shown here, but not just in the mouse model, also in the zebrafish, and even in the dog model, the golden retriever model, by data Lee Sweeney presented last year at this conference. So lots of preclinical data. My lab then wanted to get this work out of the animals into patients. So we began with men with Becker muscular dystrophy, and we showed that 1 dose of tadalafil will alleviate ischemia during hand grip exercise and restore blood flow to the arm muscles. And then thanks to funding from PPMD, we extended that to boys with Duchenne and did a dose-finding study to inform the development of this clinical trial I'm about to present. So the key question then from all this proof of concept work can improve microvascular blood flow regulation during exercise, slow disease progression, and have a clinically meaningful benefit to boys with Duchenne. So the primary hypothesis of this phase 3 clinical trial is that once daily oral tadalafil for 48 weeks compared to placebo would lessen the decline in ambulatory ability in boys with Duchenne. Oops. So this is a study design. The inclusion criteria were boys with Duchenne 7 to 14 years of age with normal cardiac function by echo with an ejection fraction of 50% or greater and a 6-minute walk distance of 200 to 400 meters on stable steroid therapy. Screening and randomization was to placebo in a lower dose and a higher dose of tadalafil with those doses based on our acute dosing study funded by PPMD. And so after 48 weeks of this double-blind study, if we had positive results, it would be an open-label extension study for 96 weeks. The primary endpoint was a 6-minute walk distance. There are a number of secondary endpoints in physical therapy assessments, and North Star assessment and time function test. We also studied the pediatric outcomes data collection instrument for quality of life, mainly rated by the parents. And then we had a number of exploratory outcomes. What I'm going to show today is the performance of upper limb, which was the priority exploratory outcome. So 504 boys were screened. 333 boys were randomized, exceeding our goal, and 315 boys completed the trial, so excellent long-term follow-up. So the baseline characteristics are shown here for the placebo group and the lower and higher dose of tadalafil, roughly equal sample size, but a little bit fewer number of boys in the low dose group. So the overall, despite randomization, a number of characteristics favored the placebo group over the tadalafil group, just in the play of chance, and especially for the low-dose tadalafil group. So if you look at the low-dose group compared to the placebo group, they were about almost a half a year older at entry. They had lower 6-minute walk distance, slower rise time from the floor, etc. And a smaller percentage of the boys in the tadalafil group were on deflazacort at entry into the study. And our own data in the placebo group, I believe Dave Cox showed the other day, shows the advantage for deflazacort over prednisone. We hope that may be helpful to the field. So unfortunately, there was no discernible benefit of tadalafil on the primary outcome of the 6-minute walk distance. The trial did not meet its primary endpoint. So from all these, what I'm going to show you, in red is low-dose tadalafil, blue is higher-dose tadalafil, and then black is placebo. So both of the tadalafil groups started out with a lower baseline 6-minute walk distance, and the curves remained parallel over 48 weeks, whether it's absolute mean values or the percent predicted. And so the change scores from baseline to 48 weeks were not statistically different for either the doses of tadalafil versus placebo. So with subgroup analysis, the boys that started with 300-meter 6-minute walk distance or higher declined at a slower over the 48 weeks, of course, than the boys that started with a lower baseline 6-minute walk distance of 200 to 299 meters. But there was no discernible effect of tadalafil in that subgroup analysis either. The North Star assessment, after 12 weeks, there was no difference in the curves. So this is a time function test that changes in velocity. There was no difference except for a tiny statistical difference between favoring low-dose tadalafil with the rise time from the floor, but this is a tiny effect. And with all the multiple comparisons, we don't think that this is clinically significant. It may be play of chance. Now, the pediatric outcomes data collection instrument rated by the parents, there was a tendency for the parents to rate for sports and physical functioning and upper extremity physical functioning for some of the tadalafil groups, but it wasn't statistically significant. And for transfer and basic mobility, it was clearly, the curves completely overlap. Now, the exploratory performance of upper limb for the overall group, there was very little decline in the pulse score, but no effect of tadalafil in the overall group. We then performed a pre-specified subgroup analysis of the exploratory outcome. And so 255 boys were 10 years old at baseline or less, so they were 7 to 10. On the left and on the right are boys that are over 10 years of age, which was only 75 boys. So if you look at the boys that are 10 or younger, there's almost no decline in total pulse score because we know that the arm disease lags behind the leg disease. But if you look at the boys that are older than 10, there's a certain indication, statistically significant, that especially that the tadalafil stabilizes the decline over 48 weeks compared to placebo. There seems to be, if anything, the better effect for low dose, and that may be because there was a higher sample size in that group in this sub-analysis. Not sure there's a real dose effect there. And then this is 72-point scale, and if you look at the shoulder dimension, it's the same direction, that there seems to be a stabilization with tadalafil, especially the low-dose group, over placebo over 48 weeks. And there are only, again, 75 boys total in this group, split between 3 groups. And again, no decline at all in the boys that are younger. So adverse events, there were very few adverse events, and they were consistent with the known side effect profile of tadalafil. There were very few serious adverse events. They were equally distributed between placebo and the tadalafil groups. As expected, penile erections increased with tadalafil in a dose-dependent fashion. But fortunately, there were no serious episodes of dangerous erections requiring treatment. And then with left ventricular ejection fraction, the left panel shows the data with echo at baseline for 24 and 48 weeks. And on the right is a 48-week change score in left ventricular ejection fraction. So there was clearly no, in the group overall, this age group that's expected, there was a small decline in ejection fraction by 1.5 percentage points in this relatively young group. If anything, there was a tendency for EF to decline less, not more, in the tadalafil group. And that's really important because Catherine Wagner's studies in patients with Duchenne in their 20s with rather advanced cardiomyopathy, there was a suggestion that there could actually be an adverse effect of sildenafil, Viagra. So we looked very carefully because of that. We didn't see any adverse effect whatsoever. So in conclusion, in the largest DMD placebo-controlled phase 3 treatment trial to date, in the first report, the performance of upper limb data, so 48 weeks of tadalafil did not lessen the decline in 6-minute walk distance or other standard measures of ambulatory ability in boys with Duchenne 7 to 14 years old age receiving a stable glucocorticoid therapeutic regimen. Had no overall effect on the upper limb functions measured by the pulse score, but it may have lessened the decline in upper limb function in boys older than 10 years of age, although the clinical significance of this remains uncertain, and this was a pre-specified exploratory outcome that would require further study. Overall, adverse events were consistent with the known tadalafil safety profile. And so moving forward, because DMD progresses faster in the lower than the upper extremities, the boys in our trial may not have been engaged in enough daily leg exercise for tadalafil to impact use-dependent leg muscle injury. This approach would only work to protect muscles against damage during a lot of use. Further studies to consider confirm the hypothesis generating upper limb data and determine if ambulatory decline in DMD could be slowed by initiation of PD5 inhibition before 7 years of age, maybe from day 1. So in closing, I want to thank the boys and their families who gave their incredible time to participate in this trial, the site investigators at 63 sites in 15 countries, and the staff for their dedication and commitment to Duchenne Connect and Treat NMD for recruitment assistance. It was invaluable. And for the physiotherapist training team for doing a really great job. And finally, on a personal level, on behalf of the steering committee, I just want to thank Dave Cox and Teresa Bauer and all the Lilly team for running a really efficient trial and for being just wonderful partners and collaborators. So thank you very much.
Thank you, Ron. It was indeed a terrific study to see in Duchenne muscular dystrophy. So our next speaker is Stuart Peltz from PTC Therapeutics, talking about the current status of Translarna. As you know, it has conditional approval in Europe, and they are working on that aspect as well as the FDA, and he's going to update us on where they are with Translarna.
Does it sound great? Well, thank you for having us. Trying to get to the other way. We're a public company, so we'll be making some forward-looking statements. Okay, great. Oops. So I just. I was just thinking I probably around our 15th meeting here. So we were probably one of the first companies to join this fight against Duchenne muscular dystrophy. I founded the company 18 years ago with the commitment to look for new treatments in rare diseases, and we've been doing that all these years. And you'll see we have a number of compounds that we're moving through the clinic with Duchenne muscular dystrophy is our most advanced program. As Pat said, we've gotten approval in Europe, which allows us to actually have a presence and be able to commercialize in multiple countries outside of the US. So we currently have offices, as you can see here, as well as the ability now we hope to have within to bring Translarna to patients in over 30 countries by the end of this year. And as I said at the beginning, we've committed to looking for treatments where there are no treatments for patients. And we've been doing this for Duchenne muscular dystrophy, cystic fibrosis, SMA, and others. We have a pipeline of a number of things. We'll be talking today about Translarna for the treatment of Duchenne muscular dystrophy for those patients who have a nonsense mutation. And just to remind you, Translarna is a small molecule. It's orally bioavailable. It's taken 3 times a day, and it's a new chemical entity with a new mechanism of action, really in a new therapeutic area, that being Duchenne muscular dystrophy, of which there are no previous treatments for the underlying cause of the disease. And as a consequence, that's why when you hear, we've been doing this now for 18 years, and there's a reason. When there are drugs in a new area, and it's a new chemical entity and a new mechanism, the regulators and the time that it takes just to get through the process is long and arduous. And that's just, I think, important for everyone to realize. And it takes not only a lot of effort from everyone here, but also the grit and tenacity of the patient community, the physicians, as well as the companies themselves. Adelor works by, for those patients that have a premature stop or nonsense mutation, it allows it to read through quite selectively to go on and make the right protein. And so that's what we're going to talk today about. And we started in 1998, and the first 5 years were to identify in the lab Adelor. And then we proved it in a mouse model. You can see where dystrophin is being able to, and I'm sure you've seen a lot of these now, where dystrophin is being able to be made and localized, and it demonstrated a reduced muscle injury. And around 2005 or so, we did a proof of concept in patients where we treated them for 28 days. And again, we were able to show pre and post treatment that dystrophin was being made in those patients. So that was the really quite exciting first time ever. Oops. This clicker's getting me in trouble. We'll go back one. Good, and the oops one. Okay. And this just shows you really the history of Duchenne, of the company and the discovery and development of it. And you can see we did the phase 2A study that proved dystrophin production. And we were left really then at the end of 2006 with talking to what's the next steps. And it became clear in talking to regulators that we needed to do clinical benefit. And there were no registration-directed endpoints for this, so we worked with the community, in particular Dr. McDonald, and that's where you heard the 6-minute walk test, and we validated and used it. But we were working in an area where there was no natural history, no understanding of what the best patient population is, what other endpoints might be good. And you can look, others have since been made. We know a whole lot more now than we did into 2006-07 when we started this. And you can see there's even guidelines written by both regulatory authorities that now tell you maybe what's the best way to do trials. So a lot has been learned in this period. You can see in 2014, what was a very important time for this community, was the first ever drug approved for Duchenne muscular dystrophy in Europe for the underlying cause of the disease, and that's where the commercialization began, and we've been working hard to get into other countries as well as into the US. Should I point this somewhere else? Okay, good. So what I thought I'd just tell you the history. So this is the first trial we did after that. We just took patients who could walk 75 meters or greater. And you can see here that the dark blue area is where 50% of the patients were between 300 or 400, but we took all comers from 75 meters to over 500 meters because we didn't know the natural history. And even in that trial, you can see we saw a 31-meter improvement over a 1 year period. So this was the first ever trial that did that, and actually a drug that demonstrated activity in a 1 year period. We learned a lot from that, and what we learned in particular was that the natural history taught us a lot. When you think, as you've heard here a lot, a dystrophin replacement therapy prevents muscle destruction. It doesn't actually, in a 1 year time, necessarily add a lot of mass or strength. So the best way to see a drug effect are in patients that are early in the disease state or high walkers. You need them declining to see if you can slow or stabilize the decline, at least in something that's a dystrophin replacement therapy. So what that other bar shows you there is we tried to, in a sense, compare. So not have kids who were so low in the ambulatory scale that there's a high risk of becoming non-ambulatory no matter what, and so high walking that you couldn't see a difference even if the drug had effect, at least in a 1 year period. So based on that, we picked an inclusion criteria. This is what, if you go back and look at the 07 data, this is what it showed. We saw a 50-meter difference. So it was based on that, and this is what we learned in the natural history, that kids who are walking 400 meters and above could stay stable more than 1 year, even multiple years. Then there's a declining time. And kids less than 300 have a high propensity of becoming non-ambulatory. And it just low, and as you've heard probably, there's about 80% fat fraction within the muscle. And so it's not that the drug can't help, but there's just, it could blur the drug's effect if there's a high degree of loss of ambulation. There might be other tests that are good. So what you really want to do is use the best population, which is looking in the range of 300 to 400. And so we had used the inclusion criteria that shows you the bottom orange, which we used previously. And we used it in study 07, but it was a small enough study that the inclusion criteria didn't necessarily represent what might happen in a bigger patient population, and that was the problem. And what you see is that the bottom line, while we wanted to include more patients that were likely to decline, you actually can see we got a wider range than we did, and in particular, we got a large number of patients that were above 400 meters. Fortunately, we had 99 patients that were in the 300 to 400 group, and because of the results that were coming out during the execution of the trial, in terms of the MRI and the loss of ambulation, the chance of that, we pre-specified that in our statistical analysis plan to look at that as an important endpoint as well. And this just shows you the data. And in this trial, because of the larger patient population, and this is the overall population on the left, we saw a 15-meter improvement. But in the pre-specified 300 to 400 group, we saw a 47-meter improvement. And it's not that the drug is, the issue is where the test is most sensitive. And what we've learned is that the 6-minute walk test is most sensitive and reliable in a narrower range of patient population. And that's one of the key learnings. And you can see what's interesting, we see about a 47-meter difference. This population was driven by the same one I showed you the previous inclusion criteria, which we saw 50 meters difference. And so what you can see is that there was a consistent benefit, while in the overall populations we saw an improvement in the 6-minute walk test of either 31 or 15 meters, but in the 300 to 400 group, whether you look in study 07, the first, or the study second trial, you see a 47-meter improvement. So quite significant and quite reproducible improvement. And this is a pretty large and important improvement. What's also important is what you'd like to see is not only results in the test, and this is from a regulatory perspective, not only in the primary endpoint, but also in other endpoints. So these are tests that you know are done in the clinic all the time, the 10-meter run, walk, the stair climb, the stair descent, and I'm sure you've heard discussions here that these are predictive of the subsequent events that occur downstream. And I think what's important here is that whether it's study 07 or study 020, you see reliable and significant improvements on Translarna versus placebo. And so what you can see is anywhere from, in the overall ITT population, anywhere from a 1.5 to 2.4 second difference. And if you put it into perspective, the clinical relevance is around a 1 second difference. And so you can see that, and the percentages really are the percentages that you see versus those who weren't on Translarna. So there was a relatively significant change in the overall percentage of preserving their ability to maintain on this test. Even larger differences are seen if you look at the 300 to 400 meter group. There's you can see really up to over a 4 second difference in the stair descent, but very substantial differences in terms of preserving function. And when you think about that, these tests are seconds, or when you look at them, what you really try and think about is at least is what I think about, is the preservation of functions, because we all know these functions as you have them. When you lose them, you don't get them back, and the predictors for loss of other functions. And you can see here that in the overall population, both in stair climb and stair descent, patients preserve the ability to go up and down steps, which I thought was very important. And also, if you look at, again, a preservation in terms of ambulation, while the 300 to 400 group have a high degree of loss of ambulation, and in the placebo group, 50% of the patients basically lost ambulation in that group. Even there was numerically improved. What I thought was quite striking, though, was in the 300 to 400 group. I should say, none of the patients in the greater than 400 meters lost in a one-year study. In the 300 to 400 meter group in the one-year study, you could see while 8 to 9% of patients lost it in the 300 to 400 group, and it was really quite consistent whether a study was 7 and 020, none of the patients lost ambulation. That's 0 out of 69 in that group lost ambulation. So a significant preservation, and that actually perpetuated more than 1 year. The other point was we did measure the North Star assessment. And this is an assessment that it measures 17 different assessments, and you get a particular score. And the score is interesting, but it doesn't necessarily, it's a combination of them. And the way it's scored is, you can get a 2 if you perform it well, a 1 with difficulty, and a 0 if you can't perform it. And so you get the score and you don't necessarily know what it means. But if you look at this, and we were talking with experts in the field, particularly a number of people in Europe as well as Greg McDonald, to say, you know, if you can perform it with difficulty, it can be subjective. But if you lose a function, that's actually a hard endpoint. And so let's count the number of functions that were preserved. Gotcha. And what you can see here is that, and we did that, we measured from easiest to hardest, and what you can see very tellingly is that no matter from simple to hardest, Translarna or Adalimumab preserved function in all of the particular functions, whether easier or harder, and was even greater in the 300 to 400 group. And you can see there was a 31% reduction in the risk of loss of function in these studies. So you can see, I mean, we have, over the years, have done a number of large studies. In clinical trials, we have well over 200 patients on commercial drug. It's also being studied in Translarna for cystic fibrosis. So we have large and long-term studies that we know not only the efficacy, but also the safety of this drug to move forward. It's shown to be a generally very well tolerated and with a very favorable safety profile. So as I said, we have it approved in Europe. We're now selling in over 24 countries. We plan to expand. We've had difficulties with the FDA in terms of getting our say in court. We're in discussions with them now. And so we were not happy with the decision and are working hard to change this decision process. And that it's obviously going to be an iterative process that we're going to be going through to work with them to work to change this decision. And so that's really what I can tell you right now where we're at, and we're highly engaged in this process. We've obviously made it our cause lab to work hard to try to do our best to bring it not only to patients outside of the U.S., but also to the patients who we've known over the last couple of decades to make sure that these families can get it too. And the patients, as I say. And so with that, let me stop there.
Our next speaker is Diane Escobar. Diane is with Akashi, and she's here to talk about HT100. Thank you, Beth, and thank you for having us, giving us an opportunity to give you an update of the program. And the backwards is forwards, right? That is the right. So for those that do not know us, I heard there is 60 new families here today. Akashi is a small biotech company owned by parents of kids with muscular dystrophy. We are in Cambridge, and our focus is to find drugs for Duchenne muscular dystrophy. We have 3 drugs in our pipeline, and the 3 drugs have as initial target Duchenne muscular dystrophy, and they are complementary, so we're not going towards putting dystrophy back. The first drug, and the one I'm going to talk today, is HT100. This drug is anti-inflammatory, anti-fibrotic, and improves muscle regeneration.
And we're going to talk more about this. The drug has been in a phase 1b to a program for the last couple of years. And as I'm going to show you, the drug improves muscular strength from baseline in these patients. Akashi has partnered with Grunenthal for Europe and Latin America drug development. The other two drugs, DT200 and A3300, target other pathways. One is a muscle building agent and the other one calcium homeostasis, and those two are going to be discussed tomorrow in the afternoon session. So let's talk about HT100. Why do we care about fibrosis? And I'm sorry about the scientists and industry people in the audience, but I'm going to talk to the parents. Is this a pointer? Ooh. Okay. So, yeah, that doesn't work. Okay. I'm going to talk with my words and with my hands, which for an Argentinian person is very difficult to do. Usually here. So fibrosis is that red stain that you see in there. On the left side of this slide is sort of a normal muscle that you can see in black are the muscle fibers, and they are sort of touching each other. And on the right side, you see all that red scar tissue that surrounds each one of the muscle fibers. That is a scar tissue, is a physical barrier, okay, that separates the muscle fibers. And if you think about it, the way that you produce muscle force is by each one of those muscle fibers connecting tendon to tendon. They are tiny, tiny and long. And then by each one connecting to each other. So they transduce the force this direction and that direction. So when you separate them with the scar tissue, that force transduction doesn't occur. This scar tissue also deranges this normal architecture of the muscle. And the main effect of that is the decrease in force production. So you decrease the force production, you decrease your muscle strength. You decrease muscle strength, you decrease motor function, right? So that's what happens in Duchenne. So what is HT100? HT100 is a molecule that binds to that enzyme, to that protein with a very long name. What that protein does is a transporter. It basically takes building blocks for making proteins and takes them into the protein-making machinery in the cell. So when the drug binds to that transporter, it doesn't let the transporter take those building blocks. And the body says, I don't have enough protein, I'm starving. And what that does is trigger a normal reaction that our body has to maintain homeostasis, to maintain balance. So it decreases all the abnormal processes that are taking too much energy and let the body use the energy for normal processes or for healing. So not surprisingly, this drug acts in areas of abnormal processes, abnormal fibrosis, abnormal scar tissue, and improve the processes to heal the body, like increasing muscle regeneration. Okay? So it doesn't act in one pathway, one target, like you heard all day today. It basically acts on this multi-pathway that is very well orchestrated, trying to create balance and homeostasis. Pretty cool mechanism, actually. So, in other words, HT100 tips the balance. On the left side, you have the diaphragm of a mouse with muscular dystrophy. It's an old mouse, it's like 9-month-old. And as you can see, a lot of red tissue, which is scar tissue, very little green tissue, which is the healthy muscle fibers, or muscle fibers, maybe not that healthy. And on the right side, you see what happened after 2 months of treatment with HT100, or halofuginum. The scar tissue basically starts to disappear, and that gets replaced by muscle, okay? So if in DMD muscle, you have muscle degeneration because of a lot of inflammation, necrosis, and fibrosis, and poor regeneration because the fibrosis decreases muscle regeneration, what HT100 does is tips the balance to the other side, all right? So let me give you an update of where we are. That's as much as I'm going to say as a background. So the HALO trials are 3 trials. HALO DMD-01 was a single dose followed by pharmacovigilance review, and then 1 week of dosing, and then another pharmacovigilance review, and then 1 month of dosing, right? And it was a dose escalating trial. And when the patients have gone through all that, they immediately were enrolled into the MDO2, which is an open extension study that lasted for 12 months. And when they were done with that, if they wanted to continue in the study, they rolled into the MDO3, which is an open continuation study. So the same patients went through the 3 trials. The patients that were included in the trials were ambulant and non-ambulant patients with any mutation. They had to have a confirmed DMD diagnosis. They could be age 6 to 20, and they had to be steroid treated on a stable dose for 1 year before getting into the trial, and they were not allowed to change any steroid dose for the remaining of the study. But they also could be steroid naive. We just did not enroll any steroid naive patients, so our patients were all on a stable dose of corticosteroids. So this is basically what the study was. You will receive, each color is a different dose, and the yellow one is the lower dose, then the green went to the second cohort, higher dose, red was the third cohort, in purple the fourth cohort, and there was a plan of possible fifth cohort that we could have enrolled, not necessarily higher, could have been lower, same different regime, so a lot of flexibility there. So there was the single ascending dose. We started with the multiple ascending dose phase of the study, continued by the 12-month extension, and continued with this open continuation study, okay, indefinite duration. Important to know that to go from the single dose to the multiple dose of the same cohort, or to go from a single dose to the single dose of the next cohort, the whole safety data was reviewed by a pharmacovigilance team, and if everything looked good, then that opened the doors to go higher and to go longer. And each step of the way, that process kept happening, and the review of the data was not just of that cohort, but of the combined data of everybody that was in the study. So this is the continuous exposure that these patients have had. By cohorts, cohort 1 on the bottom, cohort 4 on the top. And as you can see, when we talk about safety and drug activity, we have patients that, we have 23 patients that receive at least 1 dose of the drug, and they were dosed in cohort 4 less than a month, but in cohort 1 to 3 from 10 to 11 months to 18 months, okay, continuously. So the program's status update is that the program is on clinical hold, as many of you know, as of January of this year, due to a tragic cohort 4 serious adverse event that resulted in the death of a patient. The dosage was immediately suspended, the cohort 4 was terminated, but the DMD boys in cohort 1 to 3 continued to be followed per protocol. Our primary goal, of course, is patient safety, and we do feel confident that we will have a path to allow the program to move forward safely. We are targeting end of July to submit a complete response to the FDA, and we will have a dialogue with the agency anticipated at the end of August. And until that happened, I cannot share today the details, but what I can share with you is what we've been doing over the last 6 months. We've been extremely busy, and some of the safety and drug activity data that we have gathered, given the opportunity to look at all this. We have conducted an exhaustive investigation to understand what caused this fatality. These have included assessment of more than 50 biomarkers of the diseased patient and all the patients on the study, over 100 laboratory experiments, in vitro and in vivo, and we have commissioned external independent experts to review the findings. We also have done a full analysis of the safety and drug activity in the earlier cohorts, the safety, of course, of all the cohorts and activity of the earlier cohorts that have been dosed from 11 months to 18 months, on top of course, all the interim safety analysis and efficacy analysis that we presented some of that last year here. And we, of course, are going to use the finding from this investigation to inform a very conservative clinical management plan the agency agreed to resume dosing for the patients that had safe and well-tolerated long-term therapy with HT100 for so long. We want to assure the patients that participated in cohort 4 that we will provide options for these patients to resume dosing. And of course to continue HT100 with new patients. So let me talk a little bit about the preliminary results. When we talk about results, the safety results that I'm going to discuss are in 25 patients for the 4 cohorts that has been exposed from less than a month to 18 months. The mean age of these patients is about 10.6, 11 years of age. All of them on steroids, 60% ambulant, 40% non-ambulant at the onset. The efficacy data is going to be in 60 patients from cohorts 1 to 3 that has been dosed from 10 to 40 mics per kilo per day for 11 to 12 months. And you can see that the mean age is also around 11 years of age, all in the steroid 60% ambulant. And we have an external control, which is the placebo group of a synergy randomized placebo-controlled trial that was performed a while ago and published a couple of years ago. It's a pentoxifilin add-on study. We use that placebo group because these patients have been on corticosteroids for more than a year before entering the study, all through the study. The methodology of the trials are very similar. The cohort overall is a little bit less involved, which makes it a very conservative comparison if you want, because that cohort will be expected to deteriorate a little bit less than ours. So it's a not exact match, but it's a close one. If in any case, this will be a conservative one. So the preliminary results, let's first talk about the serious adverse event in the cohort 4 patient. This patient, it was a non-ambulant patient. He presented after 2 weeks of dosing in the 1-month part of the DMD-01 study with abdominal pain, hypotension, so decreased blood pressure, increased white count, and renal insufficiency. He has been having nausea and vomiting intermittently for the previous 2 weeks. He developed blurred vision, abdominal pain the day before coming, also muscle joint pain, and then came to the clinic. He was admitted that day with a diagnosis of sepsis and vasogenic shock. And after some improvement in his ICU state, the patient deteriorated and succumbed to secondary to complications. It was 3 tragic weeks. And the investigation, as I said, of this SAE is still ongoing. Let's talk a little bit about adverse events. I'm just going to give you a brief background because we talk about adverse events, but those of you that have not participated in trials probably don't understand very well what we talk about. So anything that happened to a patient that is enrolled in a clinical trial from the time that they signed it from consent is considered an adverse event. So if your kid just signed it from consent and is taking the elevator and is going to have his 6-minute walk test and trip and falls, that's an adverse event that is reported and is counted. So if the patient has taken 1 dose of the drug, even 1, then that's called a treatment emergent adverse event, okay? And once those adverse events are entered, somebody says, okay, is this related or not related to the study drug? And of course, there is a level of certainty that goes with that. So the investigator can usually decide that, and they can decide it's not related, is unlikely related, is possibly related, is probably related, or is definitely related, okay? And of course, all that can change as you learn more about the drug, so that is based on an educated guess. And of course, you have to say, mild, moderate, severe, it has a very good definition. So, having said that, we have 307 treatment-related AEs, but of those, more than 80% were mild, they resolved without sequelae, and only 30% were deemed possibly related to the study drug. So there was no definitely related or probably related AEs in this study. And then this is if we would have stopped there, that would be a very clean safety profile for the drug. We have 17% that were moderate. That will mean, a headache that needs medication. That will be a moderate AE. 2 minutes? 3 minutes, okay. And 1% were severe, 2 of which were not related to the study drug and 2 possibly related. So this basically tells you sort of the same thing. As you can see, the relationship to the study drug on the left side. In purple is the non-related, most of them non-related. But interestingly, the possibly related in red, you see an increase as the dose increase in the cohorts. On the right side, you can see the severity. Most of the AEs were mild, and you have a few moderate and a couple of severe adverse events. The possibly related AEs were mostly gastrointestinal and neurological, and these were nausea, vomiting, abdominal pain for gastrointestinal, some diarrhea, and headaches for neurological. And those, the gastrointestinal disorders, as you can see in blue, definitely increase with increase of the dose. So, these are the gastrointestinal disorders. We have a total of 102. These are all of them, but there was the possibly related. From these 102, most of them, 92, were mild, and 62, only 61 events of this in 15 samples were deemed possibly related. And if you look at the possibly related, they did increase as we increased the dose. So a very clear pattern of the tolerability of this drug has emerged. We suspected this from toxicological studies before, and it's mostly GI events and headaches. When the drug is not tolerated, that's what happens. So let's talk about drug activity in 1 minute. We are looking at muscle strength for the reasons that I mentioned before. It's the direct correlation of muscle derangement. In DMD boys, the percent predicted knee extension, for example, was 40% of control in boys at age 6 and declined to 10% at age 9. When you look at elbow flexion, you can see also very low muscle strength in young boys that really decline during the time that we follow these patients. And muscle strength has been identified by the FDA as a biomarker, acceptable biomarker, and outcome measure for accelerated and full market approval. And this is what we're seeing in muscle strength. When we put the cohorts 1 to 3 all together, that has been treated for 11 to 12 months, you can see that after 6 months there is a clear separation from their natural history and external control. And there is an improvement over their own baseline of almost 6% of muscle strength. And this is on top of the steroids, okay? Just keep that in mind. When you compare this to the external control, there is more than a 15% separation. But something to keep in mind is that we're seeing lines going up. We're not seeing 2 lines going down and one going lower than the other. We're really seeing a separation here. When you look at the muscle strength of the lowest doses of cohort 1 and 2, even at the lowest doses, you see significant improvements in muscle strength, and that really separates from the external control. We look at elbow flexion strength, and we did this because, of course, it's essential for activities of daily living, such as feeding and self-care. It's very important for non-ambulant patients. And it was just identified by a paper by a multi-group here in the US, showing us one of the simple endpoints that could be an outcome measure for non-ambulant patients. So we're going to see how that featured in our population. And what you can see is that when you look at our entire population of ambulant and non-ambulant patients, they have a significant improvement of elbow flexion strength compared to our external control. And this was, almost a 15% over their own baseline. And when you look at just the non-ambulant patients and compare them with a similar non-ambulant group of the external control from the economy paper, you can see a 22, almost 30% improvement in elbow flexion strength in the non-ambulant patients. And that is almost a 50% difference with the external control. Finally, and I have 2 more slides. We look, and I don't have time to show other data right now, but we did look at biomarkers of drug activity. So is this muscle strength because the patients are trying harder, or is this real, right? I mean, that's what everybody wants to know. And for that, we need to know that the drug is working. So we are looking at blood biomarkers that show us if the drug is really affecting fibrosis, which is what we thought that it should be doing. And what you have on the left side are markers of collagen formation and degradation, collagen 1. And on the right side, in the upper panel, the C3M, is the inflammation-derived collagen deposition. And the IC6 is muscle restructuring. And I think that it's very easy to see that the collagen 1 formation and degradation are dramatically coming down and staying down with longer treatment. When you look at the C3M, you see a decrease, an increase, and then a decrease. These patients were on clinical hold during that increase phase. So when they took the drug, it started to come down. They stopped the drug, the inflammation went up. They restarted the drug, everybody comes down again and remains low. And that's your anti-inflammatory effect. And then finally the IC6, I think that you can see a trend of increase in the IC6, which is related to muscle regeneration. So this is encouraging results. So in summary, HD100 has been safe and well tolerated at low doses ranging from 300 mg a day to 15 mg a day, and after continuous exposures for 10 to 18 months. One fatality has occurred in the highest dose group and is currently under investigation. The most common treatment emergent AE were GI, nausea, vomiting, abdominal pain and diarrhea, and neurological headaches. Preliminary data from the lowest dose cohorts, cohorts 1 and 2, shows an improvement in muscle strength over 20 to 24 months in the study and after 12 months of continuous dosing. And this is on top of corticosteroids. Both ambulant and non-ambulant subjects show improvement in muscle strength. The muscle strength improvement, and I could not show you this data today because of time, but it's accompanied by improvement in related motor function in the individual patients. For example, when we see an improvement in one patient of elbow flexion, that patient also showed an improvement of the elbow domain of the pull, or at the D2 domain of the MFM, which are our functional measures. Okay? And those improvement has also been noticeable by the parents and the clinicians. And finally, the biomarkers of fibrosis, inflammation, and muscle regeneration show clear signals of drug activity with continuous dosing. So we want to, really thank all the families and parents that participated in this study, our investigators and their teams that has been wonderful, our pharmacovigilance team members, they have a hard time. And most of you sitting on the audience that are participating to some of all these various foundations and other foundations that are funding our studies. Thank you very much.
Thanks, Diana. We are standing between you at lunch. I don't know if someone has a pressing question they'd like to ask quickly. If so, run up to the microphone and let's do it. Any questions?
Dr. Muscular, if all goes well with your conversations with the FDA, when do you envision being able to start, redosing any of the cohorts?
Yes, I don't know. Is my short answer. So we're going to have these conversations with them at the end of August. We're going to be able to share more details after we have that, and I think we're going to have a much clearer understanding of what the timelines are going to be.
Other questions? I'm going to end with one. Ron, you mentioned in the tadalafil study that in the 6-minute walk test, one of the questions or one of the ideas about a PDE5 is that you have to move muscle. So could it be that the individuals who participated in the ambulatory study weren't walking sufficiently to uptake that tadalafil and see a benefit, or potentially see a benefit?
Yeah, exactly, Pat. So I think, in the animal studies, most of the work that was done for the chronic studies were started from birth, even some before in utero. So, it suggests that I think I don't want to give false hope, but there could be, I think there's more exploration to see whether starting from 4 years of age, the first of the diagnosis, there could be some benefit when the the boys the most active. But it's, the average age in the trial was between 9 and 10. And at that time, the ambulatory activity may be not enough in daily life for the drug to get a chance to have benefit.
Thank you.